Sir, you can continue, sir.
Yes, ma'am. Thank you. Thank you. Good evening, my dear students. You're all warm. Welcome to the another session of the biological classes. Last class, what we have discussed already, the type of acute immunity, isn't it?
Yes, sir.
Yeah, further divided into two types, active immunity and natural active immunity. Again, the active immunity, what is meant by active immunity? It's nothing but the host is exposed to the antigen and antibody are produced in
The
the
antibodies will be produced in the
host
host's
body.
own body, sir.
Yeah, good, super, right. The active immunity is also a very slow process, isn't it? That things also already we discussed.
Sir.
Yeah, further divided into two types, natural and artificial, isn't it? So what
Yes,
are what are the examples? Natural means smallpox, measles, mumps. They also develop the natural active immunity. And then artificial active immunity, what are the artificial immunity? It is nothing but the
So vaccines, sir.
Yeah, very good. Exactly correct. Vaccine preparation by two proper preparations, already live or killed ones. Okay, what are the examples? Already we discussed. What is by BCG? What is by BCG? Bacillus, calamity, Gowden, isn't it? Yesterday we saw it. So these are the two types have been reported. One is called as lived one, lived one, and another one is called as killed one. Some of them are attenuated. That things also already discussed, isn't it? So you should note it down. Which one comes under the lived and which one is comes under the killed vaccines. Okay, that things also very, very important things. Okay, how will you prepare? Okay, the active immunity. And second major event is passive immunity. That things also already we discussed. It is also one of the ready-made antibodies are directly given and also to protect the body against a foreign agent. It is called as passive immunity, isn't it? That things also already discussed. Okay, and passive immunity is also further divided into two types: natural passive immunity and artificial passive immunity. Natural passive immunity, they have given which one, directly transferred from the mother to fetus through the placenta. Which one it can be easily transferred through the placenta means one of the important antibodies is called as immunoglobulin G, otherwise called as immunoglobulin G. The G denotes gamma immunoglobulin, isn't it? Okay. The milk colostrum is already what is that? After the birth, immunoglobulins are passed to the newborn baby through mother milk. Example, colostrum, first milk. Already we discussed this also. The colostrum is the first one. Second one is the second milk. It's normally conditions and also it contains a vitamin. Which one? A. The colostrum which contain vitamin A is high. It's also another antibiotics. How many months you have to provide milk? Means 3 months. You must note it down, sir. Not 3 months, maximum 6 months, sometimes 9 months, sometimes it may be 1 year. Okay, depends upon the mother. And then artificial passive immunity. Already we discussed these things also. It is also prepared from the which one hyphen immune sera. Hyphen immune sera. What is my hyphen immune sera? Some of the animals they are having hypersensitivity. Hypersensitivity of the particular pathogen is collect from the serum that is called as anti-serum. It contains antibodies. It fight against many diseases. Such type of components are called as anti-serum. They were mostly proposed mammals, particularly they prepare which one horse is an example has given. How it will be already we saw it. Okay, what are the things? See that tetanus, anti-tetanus, isn't it? again anti-rabies serum, and anti-diphtheria serum, and Garcan syrups. These are the things prepared from the, which one, hypersensitive animals. Okay, so that things also already we discussed. Already I saw it. Already we discussed it. This is a tetanus toxide given to the horse. As a result, what happened? It is produced inside the antigen and isolated from the immunized horse blood to the immune horse serum. It is also isolated from the this one. After that, it will transfer to the so many horses and also analyzed what are the changes which occurs the normal horses. It is unprotected. Why it is unprotected? Sometimes they may be dead. Okay, so suppose if it is a first immunization, sometimes it may be what is that protected. Okay, suppose sometimes it may be dead. What will happen? It will not further. It will not go the further. So we have to improve the immunizations before that in a commercial purpose as well as a laboratory oriented, isn't it? Is the process. Already we discussed it. From this one to this horse to the another one. So this is the protector horse because suppose if it is alive, okay, so it is protected. Suppose if it is dead, they will not pass on to the next one. Okay, that is a things. So already we discussed these things. So many things that were used for the production of the immunizations. One is called a spike protein. It is nothing but the outermost cell wall of the bacteria or any other pathogens. It's made up of what is that? Protein molecules. Okay, and then sometimes in the inner side is also there. Inner side of the cell wall is also made up of glycoproteins. Different types of protein molecules also used for the immunization or inoculation of horse. When you are introducing this one, what happened? The horse also undergo the inoculation process. As a result, what happened? It will be produced, okay, the antibodies. Isolated from that one, and also they may be attenuated. Attenuated in some modification also takes place, and also fortification takes place. It comes to the natural, and also this process also again and again it will be proceeded. And then, so this is also another methodology. So next, primary and secondary immune response. Already we discussed. This one is also first time exposure of the antigen. It's called as primary. Suppose if it is exposed to the second time, that is called as secondary. Okay. So tertiary means third times. Okay. Likewise. See, already we discussed these things also. Okay. This diagram last class I have explained. Okay. See, the primary immune response. Okay, when you are introducing the antibody concentration, it depends upon the antibody concentration. Suppose if it is increased the concentration, the time is also increases, but the primary immune response is very less in nature. See that curve? You can see it. This is the maximum level of the response, isn't it? Suppose it may take up to the 20 or 25 milligram, microgram like that. It may comes. Suppose when you are giving the more amount compared to the another one, take an example, primary response is this much. When you are giving the more amount and in secondary antigen exposure, what will happen? The concentration will be very high in nature up to this level. It may up to 90 or like that. So this is the maximum level of the which one secondary exposure. Okay, and also not only that, and also it is also a response is faster in nature. Why it is response is faster in nature? Because the presence already the primary immune response is also done. That's why second when we are giving, it will be very fast in nature. This is the maximum which one exposure of the antigen. Okay, these things is also already we discussed, and we will pass on to the next one is what is that? Vaccination and immunizations. This is also already we discussed. What is meant by vaccine? Vaccine is a suspension of inactive pathogen or antigen protein or pathogenic which is taken orally or injected to provide immunity for the pathogens. Sometimes they may ask the questions, what is meant by VECA? VECA is nothing but the cow. Okay, and vaccine preparation is mostly there are two types is there. One is called as live vaccine, another one is called as killed vaccines. There is nothing but the live microorganism, and also another one is called as killed microorganism used for the immunity purpose. That is called as vaccine. That is called as vaccines. This was first introduced by the name of the scientist Edward Jenner in 1796. Prepared the first vaccinations. From where, cow. Okay, and also smallpox is also discovered by the Edward Jenner. That's why, who is the father of vaccination? Means Edward Jenner is the father of vaccination. This things also already we discussed. Louis Pasteur, 1879, cholera bacteria. Okay, and then Pasteur against the rabies vaccines also prepared. These are all very, very important in the historical aspect. Just you note it down. Very, very important things. And Von Boring discovered the process. That things is also already discussed. The Von Boring is the father of passive immunization. You must note it down. Sometimes they were asked which one of the following scientists is the father of passive immunization. That things also already we discussed. Von Boring. End of the 1920, what are the vaccinations discovered? And different scientists they have given in the historical aspects. Okay. Time scale like that. Types of vaccinations: live, attenuated, inactivated, and killed one. Toxoid vaccination, cellular fraction, subunits. Cellular fraction is nothing but the molecules of made up of which one, bacteria, virus, whatever it may be. Okay, and then conjugated vaccines. Conjugated means combination of two things, isn't it? Acellular vaccination, DNA vaccines, and recombinant vaccine. Recombinant, any one of the two species, it has this ability combined together form as a new one. That is called as recombinant. Combination. Okay, these are the type of vaccine. Same one also we have to discuss. We discussed, isn't it? Here the one of the specialized one is called as biosynthetic vaccination, isn't it? This is also very, very important one. See, this one also similar to that of the previous one. So here you must note it down. These are the very important things: RNA vaccine, DNA vaccine, recombinant protein vaccine, and vectored vaccines, inactivated vaccine, and live vaccine. These are the current development. Okay, current stage. Okay, take an examples. COVID. It is also one of the RNA vaccine, isn't it? RNA vaccines. Okay. And the next vaccine history already we discussed. Okay. So these things also already yesterday we saw it. These things go to up to 1796 to until 2021. Okay. To the next one. So this is first generation and the second generation of how they will prepare an initial stage or ancient stage of the immunizations, how it will be possible. Okay. See here, this is the first stage. And live attenuated this is called as inactivated. Then is also live vaccines also prepared. That is the first generation lost to end. Second generations they prepared within the molecules with one. This is called a spikes protein. Okay, and also particle of the vaccines. Okay, they like particles similar to the whether it may be DNA or outermost core is called as protein molecule. They are used as a which one vaccination process or vaccination preparation process. And then third generation, the vectors, and then they lost one third generation RNA vaccine and DNA vaccine. Directly they were used as a nucleic acid. Okay, this is the most importantly, this is also very effective in nature. I think this is very, very important. See here, first generation, second generation, third generation, fourth generation, fifth generation. Yesterday, I asked the questions, which generation we are all living? Can you repeat me? Which generation we are all living? Which generation?
Sir, it's the fifth generation, right, sir?
Are you sure?
Sir, fifth generation, sir.
Are you sure? I am asking.
Yes, sir.
Okay, because it's mostly advanced level, isn't it? Okay, okay, okay. The first generation of the vaccines, these vaccines are prepared by the inactivating of the whole pathogens. Sometimes they may ask the question in NEET aspects. The first generation of vaccines is also prepared. How? Killed the vaccine, particularly tell us the inactivation of the whole pathogen. Whole pathogen is inactivated and also used for the vaccination process. Example, oral polio vaccine, OPV, DPT. Second generation antigenic polypeptides, it may be a protein or lipid. Okay, that combinations with the DNA molecules, otherwise called as DNA technology, were used for this one. Second, this one. Third one, DNA vaccine. So already we discussed these things from the birth onwards, how they will provide the immunization process, how it will be developed the immunological process, immune system, enhance the immune system or properties. Okay, see at birth and 1.5 years, 2.5 years, 3.5 years, 6 months, 9 months, 15 to 18 months, and 4 to 5 years. What are the things? Okay, nowadays we are using which one? We were using which one we are using? COVID first, second one, and also booster, like that we are using, isn't it? Okay. And immune disorder, we are going to discuss very deeply. This is a very, very important. You must note it down. Sometimes they may ask the questions from these persons. Improper functioning of the immune system may cause discomfort, allergy, disease, AIDS, or even death. Anaphylactic shock. What is anaphylactic shock? Sudden changes which occurs within the blood as well as the immunological system or immuno organ systems, immunological organ or immune organs. As a result, what happens now? Stroke, blood clotting mechanism. Properly, it will not function. Okay, that things is reported here. The improper functions can be divided into 3 classes. One is called as hypersensitivity disorders allergy. Hypersensitive means what is that? It is a allergic nature, isn't it? Due to some unwanted particles enter inside, number of particles will be enter inside. As a result, the immune system completely suffered. And that type of system is considered as a hypersensitive disorders, allergy. Autoimmune disorder, already you know very well, but automatically, okay, this immunological organs also same. Immunodeficiency disorder. What is the meaning of immunodeficiency? Immunodeficiency is nothing but the production of antibodies such as beta lymphocytes and T lymphocytes. Completely, the production will be lost. As a result, what happened? Other diseases they may be enter. These things already you studied in the AIDS, ELISA, enzyme-linked immunodeficiency syndrome. Okay, that is what is the meaning of that one? It is inactivated. The pathogen, when it entered into the human body, it inactivated the production of immune system, production of beta lymphocytes, and the T lymphocytes and B lymphocytes, isn't it? As a result, your immunity will be reduced. The power will be reduced. They will not fight with the many diseases. That condition is called as deficiency, immunodeficiency, due to the pathogenal activities. Harmful effect also. Okay. See, hypersensitivity disorders, allergy. Some of the example has given here, and physiology of allergy, and bronchial asthma, hay fevers, and anaphylactic shock and eczema. Okay, the hay fever already we studied. Where do you know? The pollen grain transformations. It is also lead to the some allergic effect in the branchial fever and human body. That is called as hay fever. Okay, and physiological allergy. Normally, but suppose when you are consuming any one of the dry fish, brinjal, like that, it will affect it. Physiological ability to induce the mast cell and also production of the which one histamine substances. That histamine substances it emit the symptoms. Okay, it's round like patches or redness in your skins, like that. Okay, branchial asthma already in our board. Suppose those who are smoking. If you are non-smoker, suppose somebody is smoking in the what is that? That that smoke. What will I do? Inhaling by you.
So we get cough, sir.
What?
So we get cough, sir.
Yeah. Good. Correct. So that is also another. It's suppose if it is continuously doing, what will happen? Non-smoker easily affected compared to the smokers. And hay fever already told you. Anaphylactical already told you. Eczema. Eczema is nothing but the swelling. Okay, nothing but the swelling of the a particular part. As a result, what happened now that allergy, hypersensitivity disorder. These are all are considered getting known as hyposensitive disorders. Okay. See, autoimmune disorders. Autoimmune disorders also noted down. These are the things: myasthenia gravis, Vercinius anemia, and also Hashimoto disease, rheumatoid arthritis is nothing but the swelling of joints, isn't it? And insulin dependent diabetes mellitus. Diabetes mellitus already in our but insulin dependent, some of them are insulin non-dependent, like that it is there. And then multiple sclerosis, these are all are considered in autoimmune disease. Automatically immune system will be suffered due to the some activities. Okay, you must go through it, these things. Okay, okay. And immunodeficiency disorder, particularly SCID, severe combined immunodeficiency. AIDS, acute immunodeficiency syndrome. And also third one is called as transplantation. Sorry, transplantations. These are the three headings given by the immunodeficiency disorder. Okay, so here we will discuss one by one. What are the name? Cornea transplant. You know a little bit cornea. Which part is cornea? Already know about the outermost white layer or brown layer or black layer is called as cornea. Which one is cornea? Pa? Cornea is nothing but outermost.
Sir, outermost white layer.
Very good. The
Sir,
outermost,
outermost white layer.
Good, good, good. Sure. The outermost white layer is called cornea. That cornea sometimes may be transplanted. This was first performed by in the year 1905. Was one of the first organ transplant. Which one is the first organ transplant? Means the cornea. Sometimes they may ask the questions in NEET. Okay, just note it down. And second, the first kidney transplant was done between the identical twins in the year 1954 by Charles Hoffengill. This is very, very important. First one is called as cornea. Second one is also kidney. Here, the 1905, here 1954. How many years in between these two, you know, just to note it down, 49 years. After 49 years, what they did, kidney transplant also done by some of the identical twins. Okay, that is also very, very important. And then see, types of tissues also crafting. What is my tissue crafting? Suppose, take an example. Some of the tissues are damaged, completely tear due to some accidental happen. So how it will be transferred? And you know, take a tissue from the another part of the body. You take an example, say anyone, suppose leg. From that one, you can transfer from the where, you know, in the chest or finger or whatever it may be. Such type of transplantation is called as tissue grafting, simply called as grafting. What is meant by grafting? We are substituting. Okay, substituting the original into duplicate from the any part of the body. That is called as a grafting. So there are so many types of tissue grafting have been reported here. One is called as autograft, isograft, homo or allograft, and heterograft, and successful graft order. These are the 6 have been reported in a book. So many times they were asked the question from this area. No, don't need to skip in this area. Okay. First, you should note it down. First one is called as autograft. This is the most important one and also most successful grafting. Okay, note it down. Autograft is the most successful grafting. When you are transplanting of the tissues from the one body part into another body part of an individual is called as autograft. Autograft means, suppose already told you. Suppose if you are suffering from the any part of the body in the chest, it can be replaced by the leg or any part from your body only. So such type is called as what is the autograft. And isograft. Isograft means just here, it is also another type of tissue grafting. The transplantations of tissue in between the individual of the same genetic constitutions. Example, graft between the two identical twins. Suppose you are the younger brother or elder brother. Okay, like that, you can take the tissue from your younger one. Okay, sometimes it may be a brother and a sister, like that. Okay, that is called as isocraft, genetically identical, because they are all are comes under the same genetic constitution. Such type of craft is called as isocraft, same genetical. Iso means same. Homo or allocraft. This is very, very important. See here, tissue grafting in between the genetically dissimilar individual. You also one of the mammals, isn't it? And also take an examples of tissue from any one of the mammals. Take an example, pig is also one of the mammals, and horse is also one of the mammals, and also rabbit is also one of the mammals. But different species or same species, all are comes under the heading mammalian species, isn't it? Same species, we are taking the tissue, and also it is also fixed with the human body. Take an example, the human what is a human immunoglobulin? Okay, sometimes in humulin. What is my humulin? Insulin. Okay, take an examples of insulin. The insulin is also isolated from where, Will you please tell me, any one of you? Which animals we are getting the insulin for the diabetes process?
Sir, it's a bacteria, right, sir?
No, you are preparing like commercially, that is called as a humulin. Okay, it is similar to that of the human insulin. Which one of the animals they are providing the components? They are giving. You know very about the pig. Okay, the pig contain insulin components. The beta cell produce the insulin component. That insulin component similar to that of the which one, human insulin. But we have to change the physiological alternative. Then only it will be similar to the human insulin. You have to remove or you have to add some components. That is called as alternation, otherwise called as fortification. Then only it acts as a human insulin. That condition is called as what is that? Humulin. Human insulin prepared in commercial aspect is called as humulin. Okay, likewise, I told you some of the what is that? Tissues isolated from the same species, it can be fixed with the human body. Such type is called as homo or allocraft. Another one is called as hetero or genocraft. Note it down. Once again, repeat for you. Heterocraft or genocraft. Here, what happened? Tissue grafting in between the organism of different species. Note it down. Very, very important. Here, different species means, take an examples, mammals to reptiles or reptile to mammals. Okay, reptile to bats or bats to reptiles, whatever it may be. You can select any one of the species in different species. Okay, it is not mammals. Take an example. Mammal is one of the species. At the same time, we have to choose the another one, bats. Okay, so likewise. And the last one and is called as successful graft orders. Okay, self, sibling and parents and unrelated. Unrelated means what is that? Anywhere from there. It can be fixed with the, which one, human body. That type of crafting, tissue crafting is called as a successful grafting. Take an example. Suppose if you are suffering from any one of the liver part, that is called as stem cells. Already, which is also obtained from where, your mother umbilical cord. After the birth, we will get it from the umbilical cord. It can be stored for many years. Suppose if you are suffer from that any diseases, we can use it for the development. Okay, so such type. These are all comes under the heading is type of tissue crafting. This is very, very important. Once again, repeat for you. Autocraft, most successful. Okay, and then isocraft, same genetic. Homo and allocraft and hetero and genocraft and successful crafting. These are all very, very important. No need to skip. Okay, here you must note it on very, very important things. Okay, we will pass on to the next one. See here, autocraft, patient, allocraft, other humans, genocraft, other species, alloplast, synthetic. Sometimes they were used. Suppose any one of the persons met an accident. What is that? The face is also sometimes what happened. It's something happens on it in it. Okay, we can use it the plastic. Okay, plastic surgery. Okay, already what about Likewise, suppose any one of the bone is broken, we can use another person's bone. Sometimes they were used for the animal bones also. These are all are what is a bone craft. So this is the bone craft also classification. Already we discussed the tissue crafting. Here we were discussed which one, bone crafting. Okay, these are all are very, very important things. Okay, see here. Sometimes they may ask the questions, a multiple choice question on immunology. So autocraft, see here already told you, the self, the same tissues also transfer from one place to another place of the same organism is called as autocraft. Okay, here allocraft, one person to another person, but same species. This is called as allocraft. See here, and isocraft, identical twins, brother and sisters, we can change it. Okay, this is called as isocraft. Genocraft means different species we may took from the pig and used for the human body. Okay, how it will be used? Sometimes it may be fixed. Suppose if it is not fixed, what will happen? You have to choose the another one. This is very, very important. So many times they were asked the questions. Okay, what is the next one? See here. What are the types of transplantation? Already you studied, first one is bone marrow transplantations. Where it occurs by? Anybody else? Where it occurs? Bone marrow transplantations mostly occurs in which type of diseases? Particularly? AIDS. AIDS persons suffered by the, what happened, they will not be able to produce their lymphocytes. That time what happened? The bone marrow is not used for the immunological power, isn't it? That time what happened? Replace the bone marrow from the healthy person, donor to deceased person. As a result, what happened? The donor bone marrow, they will produce lymphocytes, B lymphocytes, or team lymphocytes. As a result, the affected person, they will live for, what is that? Some more times. Suppose the person's lifespan will be 10 years after the receiving of AIDS. Suppose, imagine like that, they may live for 15 years, 5 years, it may be expanded due to the bone marrow transplantations. Okay, this is already, you note it down. And cornea transplant is already told you. Some of the persons suffered by the cornea, the donor may give, which one, cornea, we can easily transfer. But there is a specific time is that after death, you have to transfer the cornea from the healthy persons within this time duration. Otherwise, what happens? It will not be used for the further clinical aspects. And skin crafting. This is already most of the cases, burning cases, burning of skin. Okay, that cases they were doing. Suppose it is not possible, they will went for which one? Plastic surgery. Okay, got it. And then tissue grafting. This is already we discussed. What is meant by? So these are the main event mostly used for the clinical purpose. Okay, see here. Already we discussed allograft here to here. Bone graft substitute from one person to another. Sometimes they were used for the chemical also. Sometimes they were used for the plates. Okay, xenograft, other animal to human body. So already we discussed these things also. Okay. Okay. See here, skin transplantations taken from the any part of the leg to hand to leg, and leg to chest, thighs to chest, like that they may be transferred. Suppose if it is acceptable by your body, it can be fixed. Suppose if it is not acceptable, they were chosen another part, isograft, allograft, xenograft, like that. Okay, it is one of the most advantageous method, but how much they, what is that, successfully they done, we do not know. Okay, but the next one, organ transplant rejections. It is another important. Suppose one person's they are giving the heart for the open heart surgery. Suppose they may be transplant the heart. Suppose all the human bodies are accepted or not. Donor is there, recipient is also there. Donor giving the heart after the death or whatever it may be. The recipient body can accept or not. All the human body, it will not accept. Okay, Such type is called as what is that? Organ transplant rejections. It is a very important events. It's a major histocompatibility, compatibility complex. This is also denoted as MHP. Is responsible for the stimulating the rejection of tissues. The tissues it will not accept means automatically it rejected. That condition is called as major histocompatibility. The major histocompatibility is a set of genes that code for all the surface glycoprotein, because each and every animals, plants, okay, cells also made up of which one in a glycoprotein. That glycoprotein is matched with the donor, then only the tissue will be accepted. Suppose if it is not accepted, what will happen? No, it will be simply rejected. Okay. As a result, what happened? You have to choose another one. Then only which one is the compactly? That means which one is the favor for the patients. Okay. These glycoprotein mark the body cells as self and also unique for each individual. That is very, very important. This is also very, very important, because you are having, isn't it? Your glycoprotein, tissue glycoprotein is not similar to your parents. Okay, it may be differs. Likewise, your what is that? Sister also. You are having the own, what is that? Unique. That is very, very important. Unique glycoprotein molecules. See here, the glycoprotein present on the cell membrane of the donated tissue is identified to the non-selfie, what is that? T cells. This is also very, very important one, because both are similar. It is not a problem. Suppose if it is not similar, what happened? No, it will not accept the body. It completely rejected. That condition is called as what is that? Organ transplant reject. Not only that, yeah.
Sir, in such a case, the person will die, sir.
No. So suppose if it is heart, means what happened? Suppose heart is not.
Die, sir.
Yeah. So any other part like that, kidney means?
Sir, might die, sir.
It's not might die. It may take some more times, you know. For example, one more kidney is there, no? So 2 kidney we are having, isn't it?
Yes, sir.
One kidney is a failure, you know. We can replace it. Suppose if it is not possible, another kidney is also used, isn't it?
Yes,
suppose
sir.
if it is both, take an example. Nowadays the bonda money, isn't it? Affected by the 2 kidneys, isn't it? So
Yes, sir.
they may choose the donor, okay, within short period. Suppose if it is failure, we cannot use the clinical aspects. So that is very, very important. So they will not die. Suppose if it is heart, the major event is heart. Suppose if it is failure, okay, it will not replace it. Okay. So that is very, very important. Go to the next one. The cell-mediated immune reactions against the major histocompatibility antigen also. Not only that, but also antigen is also responsible for the major histocompatibility, because all antigens or antibody, it is not similar to that of the donor, isn't it? It may be differ. That is host versus graft rejection is the main mechanism of acute rejection. This is the another steps. Sometimes they may be accept, sometimes they may not accept. Already told you so many times. Likewise, the tissue also they may accept it. Sometimes they will not accept it due to the presence of which one, you know, antigen. The antigen is not similar to that of the donor, isn't it? That is also another case, another type of rejections. Some medicines such as immunosuppressant like cyclosporine. Note it down. So many times they were asked the questions. This one. Which one of the following is the immunosuppressant component? Cyclosporine. Note it down. Cyclosporine. Anti-lymphocyte globulin are given. These are the 2 important components. Mostly they used for the suppressant, immunosuppressant agent or medicine. Like that they were asked the question. Just go to the question paper 17 and 18. This question is there. Okay. Note it down. And we will pass on to the next major content or subheading is known as lymphoid organs. There are the organs where origin and or maturation and proliferation. Note it down. This is very well. Proliferation is nothing but the multiplication of lymphocytes occurs. There are 2 types. One is called as primary lymphoid organ, and another one is called as secondary lymphoid organ. So many times they were asked the primary lymphoid organ. Which one of the following is a primary lymphoid organ? Which one is not the primary lymphoid organ? Sometimes they ask the question secondary lymphoid organ. Okay, which is not the secondary lymphoid organ. Like that, they may ask the questions. See that one? What are the primary lymphoid organ? Note it down. Primary lymphoid organ where the lymphocyte originate. Lymphocytes fight against many diseases, isn't it? Viral diseases, isn't it? Suppose AIDS, taken examples of AIDS patient, lymphocytes completely reduced. Okay, into the normal level to decline. Okay, as a result, what happened? They will not fight against many diseases. As a result, all the diseases enter inside. Okay, particularly who is best friend of the AIDS? TB, isn't it? Tuberculosis is the best friend of AIDS, isn't it? So that person just go and get it the sample, get the sample from the particular persons and also analyze it. What will happen? The lymphocytes completely reduced. Okay, the originate and also mature to become an antigen sensitive such organs are called as primary lymphoid organ. Once again, repeat for you. Primary lymphoid organs where the lymphocytes originated and mature to become antigen sensitive. Such organs are called as primary lymphoid organ. Example has given here bone marrow and thymus. These are the primary. Very, very important. So suppose the lymphocytes produced from the bone marrow, this is called as B lymphocytes. Another one is called as thymus gland. Suppose the lymphocyte produced from the thymus gland, it is called as T lymphocytes. Both are primary lymphoid organ. Note it down. Very, very important. It fight against many diseases. Most of the cases, the primary lymphoid organ is activated very efficiently. Okay. See the next one. Bone marrow. So bone marrow is the main lymphoid organ where all blood cells, including what is my blood cell? Hematological studies taken examples of leukopoiesis as well as the erythropoiesis and platelets. WBC means basophil, neutrophil, eosinophil, as well as the monocytes and the lymphocytes. Okay, and also produced by the erythropoiesis. Erythropoiesis is nothing but the RBC. Leukopoiesis is nothing but the WBC. These are all are done by which one? No, bone marrow only. As a result, what happened is, see here, the bone marrow is the main lymphoid organ where all the blood cells, including lymphocytes, are produced, and it is considered equivalent to the avian brusa fabricans. Brusa fabricans is nothing but the mostly present in the bats. Okay. In our body, a bone marrow is a lymphocyte producing organ, isn't it? Take an examples of bats. What happened? A brusa fabricans is a it's act as a bone marrow in where bats, and then it is a site where the B lymphocytes maturation also takes place, where the bone marrow, the beta lymphocytes maturation takes place in the bone marrow also. This is also another important thing. See, this is considered as bone marrow. See here, the bone marrow structure here. There is a two tissues is there. One is called a spongy bone. Is also having some holes is there. And also red marrow. This is also nerve cells and blood vessels, compact bone, and also endosteum. The inner there is a yellow color is there, that is called as yellow marrow. Peristeum, periosteum, endosteum. Okay, these are all our bone cells. Once again, repeat for osteoclast or osteoblast is nothing but the bone cells. Okay, the yellow marrow is also produced which one RBC, WBC. Okay, so then only you will get the immunological power. Otherwise, what will happen now? It will be completely lose your immunity. You see that one? Spongy bones. There is a sponge in nature. Compact bone. Compact bone means it's also connected with the spongy bone. That's why it is called as compact bone. This is a yellow marrow. This is the production for the blood components. What are they? And some of them are having the red color. Red marrow as well as the yellow marrow is also present. Both are essential for the production of erythropoiesis as well as the leukopoiesis. Okay, so this is important things. Note it down. Here they may ask the questions, which one is the yellow marrow is responsible or red marrow? Okay, and also periosteum. Okay, sometimes they may ask the questions like that. See the another also, the hematopoiesis. Hematopoiesis is nothing but the blood component. What are the blood components? See here, So many things is there. This is called as stem cells, already used for the stem cells. What purpose? Any one of the organ is, what is that, affected by some diseases or something happen inside a fracture or something else, when you are placing the stem cells. Nowadays there are so many stem cell bank also is there. Likewise, blood bank, stem cell banks also present. A lymphoid progenitor cells, T lymphocytes, B lymphocytes, platelets, erythrocytes, monocytes, neutrophil, basophil, eosinophil, and also other components. These are all are produced by the bone marrow. That's why it is called as hematopoiesis. All the blood components, what are the solid particle present in it, all are excuse me, produced by bone marrow only. Okay, right. Next one is thymus. You must note it down. What is by thymus? Where it is present? What will be the main function of the thymus? These three things you must note it down. Okay. So first one, thymus is a lobed organ located near the heart and beneath the chest bone, that is called a sternum. Just imagine your sternum is there. Where is the sternum? All the bones are united in the triangle shaped bone present in the center of the chest. That is called as sternum. Okay. Behind the, otherwise called as beneath the chest bone, you are having a bilobed near the heart also. Okay. That gland is called as thymus gland. It is a quite large at the time of birth. During the time, what happened? It will be big in nature. That is also called as a large in size. After the growth, when we are growing, what happened? Automatically, the size will be reduced into small one. But keep on reducing in size with the age, and by the time of puberty is attained. That is my puberty. First, what is that? Age attaining process. Okay. That is called as puberty. That time what happened? The thymus gland size will be reduced. It reduced to very small in size. Okay. Then T lymphocytes are produced by the bone marrow but mature in thymus. Very, very important thing. T lymphocytes produced from where, bone marrow but maturation in thymus gland. This is also very, very important. So many times they were asked the questions. Where is the maturation takes place? The T lymphocytes production, where is it? Like that they may ask the questions. So don't forget this important point. See, this is called a thyroid gland. Okay. There is a trachea, isn't it? Lungs, branchial cavity here. This is the lungs, right side or left side lungs. Okay. The center, there is a space is there. This is called as thymus glands. It's covered with the which one, Which one, Sternum. The chest bone, isn't it? So just to imagine, sometimes they were asked a question, thymus gland present beneath the skin or near the heart, and both sometimes they may give. You have to give the excuse me, correct answers. So go to the next one. See the structures. This one is also thyroid gland. Just what is that? Butterfly shaped. Present the larynx below, trachea, and subclavian veins. This is called a subclavian vein. This is called a thymus gland. See here. This is the thymus gland. Then there are branches, left lungs and also right lungs. This is heart. Beneath the heart only the thymus gland presented. Here the beta lymphocyte production takes place and also maturation in thymus. Okay, go to the next one. See here also another slide for your view. This is the thymus gland. What about this one? The green color. This is called as lymphatic system, otherwise called as lymph node. There is a small, what is that? Not like structure is, this is called as lymph node. Okay, these are all our lymphatic system, completely collectively called as lymphatic system. What is the main uses of lymphatic? It's also act as a, what is that? Immunological effect. Okay, so go to the next one. Primary lymphoid organs, bone marrow and thymus provide a micro environment. Note it down. This is very, very important for the development and maturation of the lymphocytes. Micro environment, what is it? It will not produce more amount. It will produce a very least amount only. That's why they have given us a micro environments. Okay. Go to the next one. Secondary lymphoid organ. primary lymphoid organs, only two is there, B lymphocytes and T lymphocytes, bone marrow as well as the thymus gland. Secondary lymphoid organs means, what is that? The organs where lymphocytes interact with the antigen and proliferate to become effector cells. That means, what is the meaning? It will interact with the cells, the lymphocytes interact with the antigen and proliferate it to become effector cells. The antigen will be proliferated. It will be produce more number of antigens. So, okay, such organs are called as secondary lymphoid organs. What are the example has given here? No? Spleen, lymph node, tonsils, Peyer's patches. Note it down. This is very, very important. Peyer's patches of small intestine and also appendix is also one of the secondary lymphoid organ. Okay, are called secondary lymphoid organ. Once again, repeat for you. Secondary lymphoid organs. The organs where lymphocytes interact with the antigen and proliferate to become effector cells. Such organs are called as secondary lymphoid organs. Examples: spleen, lymph node, tonsils, Peyer's patches of small intestine and appendix are all called lymphoid organ. Which lymphoid organs? Secondary lymphoid organs. Okay, to the next one. See the structures. So this is the structure of secondary lymphoid organs present in our body. See, adenoids. Okay, this here given as adenoid. Tonsil, here the tonsil. This is the thymus gland. Thymus gland comes under the heading primary or secondary? Primary lymphoid organ, isn't it? See, the spleen is a secondary lymphoid organ. Okay, intestine, Peyer's patches. Okay, see here the Peyer's patches present in the intestines. Appendix, bone marrow is the, which one you know? Primary lymphoid organ. Others are secondary lymphoid organs. Okay. See here. So another one is also thymus, lymph node, bone marrow, spleen, lymph vessel. Sometimes they ask the questions. Spleen is present in where, you know, left side or right side? Left side only. Just note it down. This is called as lymph node. Lymph node is nothing but there is a knot-like structure present in the lymphatic system. This is the spleen. Okay, note it down. For the next one, spleen. Once again, repeat for you. Which one is a graveyard of RBC? Which organ is called as graveyard of RBC?
Sir, spleen, sir.
Very good. What is the main functions of the spleen? Main functions of the spleen. Will you please?
Sir. Sir, it breaks down the RBC, sir.
One, okay. What about a main function? Main one. Dhanalakshmi, Ritika. Do you have any idea about this one? What is the main functions of spleen?
No idea, sir.
No idea. Okay. The spleen is a very, very important one. All the blood microorganism, it can be filtered by spleen only. Suppose your blood contain microorganism, you know, it can be filtered by the organ is called a spleen. That's why it is called as graveyard of RBC is also there. Graveyard means nothing, slaughterhouse of the RBC. Okay. See, the spleen is an organ in the upper far left part of the abdomen. Left part of the abdomen are having the, which one you know? Small organ is there to the left of the stomach. The spleen varies in size and shape between the peoples, but it is commonly first shaped, purple and about 4 inches long. Note it down. How many inches? 4 inches long only. Okay. The large bean-shaped organ mainly contain lymphocytes and phagocytes. What is by phagocytes? Already told you. It's nothing but engulfing of the cells or cell eating process done by macrophages. What is my macrophages? Macrophages. Macro means large. Phages means it's nothing but the cells. Which cell present in a body is a very large in size? That means WBC. Take all the WBCs also comes under the heading is known as macrophages. Okay, don't forget it. So this is also very important. Here you must note it down. A spleen, where is the location, size, how many inches, what they will be produced. These are the three things you must note it down. Okay, go to the next one. Act as a filter or blood by the trapping of blood-borne microorganisms. This is very, very important. Addition with this grave add of RBC. So this is the another point. Filtering of the microorganisms. Second one is graveyard of RBC. Okay, and the third one is spleen compared to the heart. Which one is a high concentration of blood also present? Means spleen. The spleen is also serves as a large reservoir of the erythrocytes. These are the points very, very important regarding which one know spleen. You must note it down. Spleen is also a serves as a large reservoir. It gets more amount of RBC. That's why. Okay, spleen is also act as a reservoir of erythrocytes. See here. So this is the entire structure of the liver and nearest things. See, this is a liver, the red color. The below the liver there is a one green colored leaf-like structure is called as gallbladder. What is the main uses of gallbladder? The gallbladder mostly they were used. Okay, what is that? What is the use of gallbladder? They produce two components. What are the two components? Biliverdin, bilirubin. What is the use of biliverdin, bilirubin? Mostly they used for the digestion of fat only. Okay, without this component, the fat it do not easily digest. Not only that, and also lipase is also enzyme is also essential. Okay, that is all. And another one is this one is called as what is that? There is a small finger-like projection is there. This is called as pancreas. The pancreas already know very well, but there are three parts is there. One is called as three cell is there. One is alpha cell, another one is beta cell, and then third one is called as gamma cell. The beta cell secrete the insulin, alpha cell secrete the glucagon, and gamma cells when it acts, no, the both the cell is absent, both the cell activity is completely absent. That time what happen, automatically gamma cell it will be, but when I take response. Above the pancreas there is a pink color that is called a spleen. Just imagine the spleen is a very, very important. It is also comes under the heading secondary lymphoid organ. Note it down here. 4 inches. Okay, not only that, and grave order RBC, and also it filter the microorganism also. So these are all are very, very important for the spleen. Okay, and go to the next one. See here. this is the spleen. Okay, so stomach, and then pass on the next one. See here again the same diagram and also cross section also. Capsule, tubercle, vascular, sinusoid, and white pulp. These are all are the cross section of the spleen. When you are going to take the cross section of the spleen, we can see these are the what is the parts. Sometimes they may ask the questions. Okay, what is the white pulp? Where it is located? And tubercle. Okay, capsule already know. Capsule is nothing but closed circuit. There is no any way. That is called as capsule. Okay, the next one. And then pass on to the next one is lymph node. It is also another type of what is that? Secondary lymphoid organs. See here, the small solid structure located at the different points. Already told you there is a small knot-like structure present in the where a long and lymphatic system is called as lymph node. I will show you the diagrammatic representation, isn't it? Next slide I will show you. The trap the microorganism or other foreign antigen also done by which one, lymph node. Lymph node, what they will do? Trap the microorganisms or other foreign antigens. Foreign antigen also trapped by the lymph node. Okay, see here, next one. The antigen trapped into the lymph node responsible for the activation and the differentiation of the lymphocyte and cause a immune response. Not only that, it will not function as a trapped one, and also responsible for the activation and also differentiation of the lymphocyte. Different lymphocytes also what is that? Done by which one? Lymph node only, and also fight against many diseases. It's also responsible for the immune system. This is also done by lymph node. Once again, lymph node, there is a small solid structure present. The long lymphoid system is called as lymph node. What is the main functions of this one? Trap the microorganism or other foreign antigens. Not only that, and also differentiation of the lymphocytes and cause the immune response. Okay, see that lymphoid system. Okay, so already this is the lymphoid system. There is a knot is that small dot is the small round like structure is called as what is that? Inguinal lymph nodes, axillary lymph nodes, palatine tonsils, cervical lymph nodes, and cisternae cyli, right lymphoid duct and left lymphoid duct, like that they may give. Okay, so these are all the entire system of lymphoid organs. Okay, so we finish it up this well. So lymph node, see the structures, lymphoid channels. Okay, this is the channels and capsules also there. It is covered by a cells. Medulla is nothing but the middle part. It's similar to that of the kidney. Cortex is also there. Blood vessels also pass on through way. So collectively called as what is a lymph node. See the structure lymph node. They are collectively called as lymphoid system. There is a small dot-like structure. The cross section has given here. So what are the internal structures also present in it? Okay. Once again, go through it, you will get the answer. And then mucosal associated lymphoid tissues. This is very, very important. So many times they were asked the expansion and mnemonic aspects. Mucosal associated lymphoid tissues, simply called as MALT, is located within the lining of major tract. Note it down. Major, what are the major tracts? Already we are having a respiratory tract. Respiratory tract also having the mucosal membrane. Nothing but the mucous membrane associated with the lymphoid. That means the mucous membrane fight with against the many diseases. Okay, such type of component is called as MALT, mucosal associated lymphoid tissues. Okay, which one they have given as a respiratory tract is also having, digestive tract, and also urinogenital tract. Already we discussed. These are all having MALT substance. And MALT is nothing but the lymphoid tissues fight against many diseases with the mucous membrane. Okay, it constitutes 50% of lymphoid tissues in this category. Which one? Respiratory organ, digestive organ, urinogenital organ contained. How many percentage of the lymphoid tissues? 50% of the lymphoid tissues mostly present in this area only. Okay, so go to the next one. See, this is the structures. Okay, here we will not see that properly. We will pass on to the next one. You can see it. See that one? Salt, skin related. This one is called as NALT. This one is called as GALT. This one is called as PALT and MALT also. Okay, see here how many things is there? PALT, NALT, GALT, and salt. Okay, likewise MALT also there. Mucous associated. NALT, it also another one. GALT, it is also another one. I will tell you. You have to find out. This is your homework. Which one is PALT? Which one is NALT, salt, and GALT? You have to find out the answer. Tomorrow I will ask you. Okay, otherwise I will tell you. Okay, go to the next one. And here the question has given, which one of the following is the main graveyard of RBC? Already we discussed that one. Bone marrow, it is not a graveyard of RBC. Spleen is the answer. All vertebrate graveyard of RBC done by which one? One of the secondary lymphoid organ is called a spleen. Note it down. Liver, it is not one. And kidney, it is not, comes under this category. So this is the important one. And then another one is iron containing pigment of hemoglobins. Answer heme. These are all question pattern, question aspect. They may ask the question. And protein containing pigment hemoglobin. Answer globulin. Graveyard of RBC, spleen. Blood bank of in the body. Once again repeat for you. Blood bank. Spleen is also blood bank. Spleen is the blood bank of which you know body. Lifespan of RBC already 120 days. What about WBC? Okay, and platelets. That things also you must know. Platelets I think is 7 days, and WBC maximum 15 to 20 days. Okay, total count measured by the instruments known as a hemocytometer. Note it down. This is very, very important. Hemocytometer. Degrees of RBC is caused towards anemia. Suppose increasing of RBC is called as polycythemia. Note it down. These are all, or so many times they were asked the questions. Okay, go to the next one. We'll finish it up till later. And then here, the pair pages are categorized as which of the following? So lymph node. Pair pages is also comes under the lymph node. Okay, it is not comes under the branches associated. That is called as spalt. Mucosa associated. That is called as smalt. Lymphoid fluid, a primary lymphoid organ. These are all are the absence. Pair pages are categorized as which of the following means you have to tell us the lymph node only. It is also act as a lymph node. It is also one of the lymph node. And another one is what tonsils are located at the back of the throat. See here, palatine. Okay, palatine. It's not tympanic, pharyngeal, lingual, and nasopharyngeal. Which one is the correct answer? The dark one. Which one is the palatine? Sometimes they may ask the question just for the example has given here. You note it down. Okay, and that's all about today classes. Okay, remaining things we will see on the next classes. Will you please tell me what are the things today we have to discuss? First one is what is by primary lymphoid organ, what is by secondary lymphoid organ, what are the examples, where it presents, which one is graveyard of RBC, and what are the types, what is by plat, what is by clat, what is by malt, like that we discussed. Okay, so what is by lymphatic node, what is by lymphatic systems, what is the main functions of lymphatic system. Okay, that things already we discussed here. So many times they were asked the questions. Once again I repeat for you, please go through it and take a notes, those who are having the which one black letters and bold letters. Okay, that's all about today class. We will see remaining on the next class. Bye, take care.
Thank you, sir.
Thank you. Bye. Thank you, ma'am. Thank you, my dear students.
Thank you, sir.
Thank you, pa. Thank you, ma'am.
Thank you, sir. Thank you, sir.
Thank you so much. Bye.